This is a presentation about the overview of metabolism. So first things first is that we want to start with glucose, which is a basic sugar. And through a series of reactions in glycolysis, we go through glucose 6-phosphate, fructose 6-phosphate, so on and so forth, and end up with pyruvate. And as you can see over on the left side, there is sucrose, which can be used as a precursor to glucose along with fructose. And as you can see, fructose can also be converted into different molecules, which can have a different starting place within glycolysis. Next is the regeneration of glucose. So this is done by glyconeogenesis, where we take pyruvate and somewhat take glycolysis and just run it backwards. However, there are different steps that need to be taken due to the committed steps of 1, 3, and 10 in glycolysis. And on the right side, you can see that dietary fats are used to create glycerol, which through a series of reactions can be incorporated into glyconeogenesis to regenerate glucose. So lactose and galactose can also be used as a starting material. So lactose gets broken down into galactose and glucose, and the glucose goes straight to glycolysis, and galactose, through a series of reactions, eventually does become glucose, which then goes to glycolysis. Then, after glycolysis is completed, you end up with two molecules of pyruvate, where the pyruvate gets put into the pyruvate dehydrogenase complex, where it is eventually converted into acetyl-CoA, where through a series of reactions within the citric acid cycle creates NADH and 1-GTP and FADH2. Then in the electron transport chain, this is where NADH and FADH2 will be converted and they generate ATP. To synthesize ATP, the ATP synthase is used, where there are 3 different reactions where essentially ADP and inorganic phosphate are rotated, and with the help of losing water, ATP is created. We can also use amino acids to create different molecules that can be used in the different reactions previously discussed. So alanine can become pyruvate, where, as we saw previously, is the final product in glycolysis. Aspartate can become oxaloacetate, which is one of the molecules in the citric acid cycle, and glutamate can become alpha ketoglutarate, which is another molecule in the citric acid cycle. As you can see, the synthesis of cholesterol is very complex, and it takes multiple reactions to synthesize it. Finishing cholesterol, a major molecule that is noted is squalene, which through a series of reactions creates the first precursor to cholesterol, which is the first steroid, and then through a variety of different steps, cholesterol is synthesized. Methionine can also be used, and there are 2 different outcomes, where in one pathway it creates succinyl-CoA, and in the other pathway it creates cysteine. In this slide, this is showing amino acid synthesis, where the essential amino acids are being generated for our body to use to create different proteins. In this slide, we have triglycerol that is synthesized and it is used in lipid metabolism. And in this slide, it's showing the glycerol that was created in the previous slide that was used in lipid synthesis, and here, through a series of reactions, it can be used to create energy. In a previous slide, in fact, it was the cholesterol synthesis slide, we saw PRPP, which, once again, through a series of reactions, is created, and along with many other essentials that we will need for metabolism. And finally, this slide shows how not only does our body use ATP for energy, but our body also uses UTP, CTP, TTP, and GTP. The ATP and GTP are synthesized from ribose 5-phosphate, and the reason that these two are similar is because they are purines, whereas the other three are pyrimidines, which causes differences and they need to be synthesized in different ways.